toxic impurities in DMF. The first few slides of my brief talk are about DMF major deficiencies and why we focus on PGI-related deficiencies. The second part is to refresh our memory of ICH M7. We follow ICH M7 to assess PGIs in drug substance. The third part is about how we assess PGIs in our daily work. This is a DMF assignment under GDUFA 2. In a 10-month review clock, DMF assessment has about 7 months to complete. The typical response time by DMF holder is 2 to 3 months. Response in a timely way might allow for a second cycle assessment. So what can we do to reduce response time? The key is DMF major deficiencies. What are DMF major deficiencies? What are DMF major deficiencies? Lauren has shown these slides in yesterday's presentation. I'm not going to repeat it here again. One thing I like to mention is that second subcategory of DMF major deficiencies is qualification of impurities. Our division, DL API, has established a database to track DMF major deficiencies since October 2017. As of October 2018, over 30% of DMFs have major deficiencies in the first review cycle. The average response time is 86 days. The main subcategory of major deficiencies is qualification of impurities. Control and qualification for PGIs is the main reason for major deficiencies in DMF complete response letter. As shown in the graph on the right, subtypes of PGI-related deficiencies, including PGI not addressed, control insufficient, AMI or QSAR data insufficient, TDC are acceptable, and M7 exemption are acceptable. To understand these PGI-related deficiencies, let's refresh our memories of ICH M7. We follow ICH M7 to assess PGIs in drug substance. The key concept in ICH M7 is TDC, threshold of toxicological concern. TDC is developed to define an acceptable intake for DNA reactive substances that have the potential to cause cancer. High-potency mutagenic carcinogens are controlled below compound-specific acceptable limit. TDC is calculated with acceptable intake and MDD, maximum daily dosage. Treatment duration should be determined prior to selection of acceptable intake. Treatment usually can be located in ROD label. Alternatively, refer to treatment durations based on clinical use scenarios. For an individual PGI, the acceptable intakes range from 1.5 microgram per day for lifetime treatment to 120 microgram per day for treatment less than 1 month. According to ICH M7, impurity could be divided into 5 classes with respect to mutagenic and carcinogenic potential. From bottom to the top in the table, class 5 impurities have structures alerts or alerting structure with sufficient data to demonstrate non-mutagenic. Class 4 impurities have alerting structure, same alert in drug substance, or are compounds related to drug substance which have been tested and are non-mutagenic. Class 3 impurities have alerting structure and related to the structure of drug substance. There is no mutagenicity data. Majority of PGI-related deficiencies is about class 3 impurities. Class 2 impurities are non-mutagens without carcinogenic data, and class 1 impurities are non-mutagenic carcinogens. So what are the control strategies for ICH M7 classified impurities? This is the decision tree for impurity qualification. The starting point is classification of impurity. Class 1 impurity is controlled below compound-specific acceptable limit. Class 2 is controlled below TDC limit. Class 4 and Class 5 impurities are non-mutagenic. ISHQ3 applies. For Class 3 impurity less than TDC, TDC or ISHQ3, whichever law applies. For Class 3 impurity above TDC, any QSAR test could be carried out. If QSAR prediction is negative, the impurity is controlled as Class 5 impurity. If QSAR prediction is positive, AMI's test could be followed. If AMI test result is positive, the impurity is upgraded as Class 2 impurity. Alternatively, Class 2 and Class 3 impurity could be controlled at the early stage in the manufacturing process. Option 3 or option 4 of ISH-M7 could be selected. There is one exception for companion specified impurity. Companion specified impurity overrides the structural law unless or a structural law of very high concern, such as N-nitrosol compounds. ISH-M7 also provides 4 control options for Class 2 and Class 3 impurities. Option 1, monitor the impurity in drug substance. The acceptance criteria is below TDC. Option 2, monitor the impurity in intermediate starting material or in process control. The acceptance criteria is still below TDC. Option 3, monitor the impurity in intermediate starting material or in process control. However, the acceptance criteria is above TDC. Understanding of impure fate and spike purge batch data are required. Option 4, design robust process controls to reduce the risk of impurity to negligible. The risk assessment could be submitted as purge factor for the clearance of impurity by manufacturing process. Option 4 is especially useful for those impurities that are extremely unstable or for those impurities that are introduced early in the manufacturing process can be effectively purged. So, how do we assess PGI in our daily work? This slide summarizes reviews checklist on the task. Prior to assessment, step 1, confirm whether M7 exemption is applicable. Step 2, locate treatment duration and MDD in ROD label. Step 3, identify acceptable intakes and calculate TDC. During assessment, identify alerting structure based on synthetic route. Step 2, search internal QSAR database and submit internal QSAR requests if needed. Step 3, submit Pharm Tox Council. If any test results has been provided in the submission, the internal turnaround time is about 3 months. Step 4, submit Pharm Tox Council. If QSAR report has been provided, the internal turnaround time is about 2 weeks. If control option of ISH-M7 has been adopted, the assessment is immediate. This is a synthetic route of drug substance extracted from literature. In order to identify structural alerts, the synthetic route has been expanded to include reagents and all in-situ intermediates. Intermediate B1, intermediate C1 is also an in-situ intermediate. It is an alkyl ester of sulfonate. Intermediate D is also an epoxide. In addition to structural alerts, we also pay close attention to the potential risk of reagents, such as methyl sulfonyl chloride or harsh reaction condition, which might generate PGI. In this case, reflux of taurine sulfonic acid isopropyl solution might generate PGI isopropyl taurine sulfonate. Without compromising drug substance quality, we recommend selection of best control option to minimize assessment time. Intermediate B1 is introduced early in the manufacturing process. It consists of stability, chemical reactivity, and solubility. Option 4 is recommended. Discussion of purge factor for the clearance of the impurity by manufacturing process is required. Intermediate C1 is controlled by IPC. Option 3 is recommended. Spike purge batch data are required. Intermediate D has an alerting function group of epoxide. The epoxide ring has 3 substituent groups and the reactivity is very low. If predicted to be negative for bacterial mutagenicity by QSAR analysis, intermediate D could be controlled as regular impurity. ICH Q3A applies. Methane sulfonic chloride reacts rapidly and completely with water. Option 4 is recommended. Scientific justification should be sufficient. The last one is the isopropyl taurine sulfonate. Isopropyl taurine sulfonate is generated at last stage. Option 1 is recommended. Test exclusion is allowed if sufficient batch data are provided. At slide number 4, we list 5 subtypes of PGI-related deficiencies. Here's what industry can help to avoid these deficiencies. For PGI not addressed, we recommend a complete list of PGIs from reagents, starting materials, intermediates, or byproduct through simple comparison with a known alerting functionality and through searches of published information such as CCR's database. Control not sufficient. Control not sufficient usually occurs when option 3 is selected. Because acceptance criteria is above TTC, demonstration of process capability at the proposed level is required. Regarding AIMS or QSAR data not sufficient, AIMS tests should be performed per ICHS2 and OECD 471 guidelines. QSAR analysis should be also carried out with ICH-M7 compliance software. For TTC not acceptable, the likely cause is incorrect MDD or incorrect treatment duration. If MDD or treatment duration cannot be located in ROD label, communication with your under applicant or submission of controlled correspondence is highly recommended. The last one is M7 exemption not acceptable. M7 exemption applies to drug substance and the drug product intended for advanced cancer indications, or drug substance itself is genotoxic at therapeutic concentrations. This slide is about post-approval changes. Control of PGIs doesn't stop after DMF becomes adequate. Post-approval submission involving the drug substance CMC change should include an evaluation of potential impact associated with mutagenic impurities. Specifically, changes should be evaluated to determine if the changes result in any new mutagenic impurities or higher acceptance criteria for existing mutagenic impurities. As you are aware, for certain related recall occurs in July 2018 due to the presence of N-nitrosyl dimethylamine, NDMA in API. If a manufacturer detects new or high level of impurities, action should be taken to prevent changes to the product's safety profile. In summary, control of PGIs is one of the main reasons for major deficiencies in DMF complete response letter. Following ICH M7 step by step could make control of PGIs right the first time. In-depth understanding the formation, fate, and purge will facilitate selection of appropriate control strategies for PGIs. This will reduce assessment time and increase the chance that DMF becomes adequate within under first cycle review clock. The last take-home message is compliance with ICH M7 is required in post-approval submission. please refer to ICH M7 Appendix 1 for the applicable scenarios. That's all I have. I'm going to turn the podium to Vicky. Thank you.
Thanks, Hong Bao. All right, hello, I'm Victoria Keck. I'm a toxicology reviewer at OGD. I am one member of a very small but mighty PharmTox review team that's nestled within the Division of Clinical Review, which is within Office of Bioequivalence at OGD. And I'm excited to be here today because this is actually the first time that the PharmTox team has participated in the Generic Drug Forum. And I think this is a really great opportunity for us to do some outreach and potentially help you with your generic submissions. Now, I only have 14 minutes, and if you get nothing else from my talk today, what I would like you to take home is to please go check out the Good ANDA Submission Practices Guidelines. There's a lot of great information in here for many different aspects of your ANDA submission, but particularly with PharmTox issues, really common PharmTox issues related to impurities. You'll find some great tips in there. All right, so what does the PharmTox team do? We conduct generic formulation safety review with the expectation that your generic formulation has the same safety profile as the RLD. And as such, your impurity profile should not pose a greater risk than the RLD. We conduct a compound-specific safety review according to the methods that are outlined in ICH M7 and ICH Q3A and B, as Hong Bao had talked about. And again, the FDA Good ANDA Submission Practices is good for you to check out to see our approach of conducting these reviews on impurities. So I've put together two cases for you that illustrate two common scenarios we see where we have some issues with justifications for impurity limits from a PharmTox perspective. And the first case is about use of in silico methods to characterize the safety of an impurity. And in this case, we're looking at ICH M7 and ICH Q3A drug substance impurities because we have a drug substance, a DMF, that's currently being referenced by a pending ANDA for an oral chronic use indication. The maximum daily dose is 1,000 milligrams. In this drug substance, there's an impurity we'll call impurity A, and it's currently exceeding ICH Q3A limits. They're proposing a limit of 0.5%, but since it is being referenced by an ANDA that has a chronic oral use indication and the maximum daily dose is 1,000 milligrams, these are the limits that would apply. For any potentially genotoxic impurities, the limit should be 1.5 micrograms per day. And for any non-mutagenic impurities, the limit should be 0.15% or 1 milligram per day, whichever is lower. So the applicant submitted in silico prediction to address mutagenicity as well as some general toxicity endpoints. So the question is, when are in silico methods adequate for characterizing the safety of an impurity? And hint, you can check the Good ANDA Submission Practices Guidance. We actually talk about this in this guidance document. So when are in silico methods adequate? I'll tell you. For prediction of bacterial mutagenicity, that's when they're adequate. Submit one expert-based and one statistically-based model, and please submit the full study reports. We actually read the entire full study report. And you can see the ICH M7 guidance for further information about bacterial mutagenicity. So common pitfalls we see with QSAR submissions, we often see just a single model submitted. Again, we need one expert-based and one statistical-based model. Sometimes it's a software we're not familiar with, so we have insufficient information on model validation, so please submit that. Also, full study report not submitted. I'm going to keep on repeating this. Full study report, we need to see them. And I have a side note here, Kramer classification. Very often, in conjunction with these in silico predictions, we're seeing Kramer classification being relied on to justify your limits. And this is considered insufficient to characterize safety because it's based on classifying compounds by their structure, and the methods are not validated for the endpoints that Kramer classification is predicting. It doesn't really cover all of the endpoints that we look at for drug safety review. So when an in silico method is not adequate, when it's not bacterial mutagenicity prediction. General toxicity is not validated for the endpoints of general toxicity studies. Do not rely on them. So what is acceptable to address the general toxicity of your impurity that's above ICH limits? You can either conduct a repeat dose general toxicology study. Please conduct this within the context of use of the drug product. So if it's an oral product, an oral study, intravenous study. And you can provide published literature, and please provide, again, the full report of your study or provide the article that you are referencing. So in review, we had a drug substance that's being referenced by an ANDA with oral chronic use indication. Maximum daily intake of an impurity in this drug substance exceeds ICH-Q3A qualification threshold. The applicant submitted in silico prediction to address mutagenicity, and in this case, it was acceptable. They submitted the full study report. They had one expert-based and one statistical-based method, and we told them that they were allowed to control up to ICH-Q3A limits. Now, for the other endpoints that they submitted for in silico prediction, we did not accept those. So we told them, in order to characterize the safety of an impurity above ICH-Q3A limits, you need to provide repeat dose toxicology study that covers the context of use of your product, and/or you could submit published literature, or you can conduct a comparative impurity analysis with the RLD. Case 2, metabolite justification to control an impurity above ICH limits. I think there was a question about this earlier today. For this example, we're going to look at a drug product, so ICH-Q3B limits apply. This is, again, another oral drug product. The maximum daily dose is 1,000 milligrams. There's an impurity B that exceeds ICH-Q3B qualification threshold. They're proposing a limit of 3%. However, because of this product's maximum daily dose, the ICH-Q3B limit would be 0.2% or 2 milligrams per day. The applicant submitted a metabolite justification using public literature, and they claim that impurity B is a metabolite of the active pharmaceutical ingredient. Is this justification adequate? You can actually check the Good ANDA Submission Practices Guidance. We talk about this in this guidance document. So is it adequate? They did provide published literature, and it did show that this impurity is in fact a metabolite that is detected in rodent and human urine. However, there is no quantitative information about systemic exposure to this impurity as a metabolite. So we don't know if there's enough presence of this impurity as a metabolite that would meet or exceed levels that are being proposed as a limit as an impurity in the drug product. So what is an adequate justification? We want to see quantitative information on systemic exposure. We need to see plasma levels that either equal or exceed the proposed clinical exposure levels of your impurities limit. So demonstrated systemic exposure at a level to support the impurity limit that you're proposing. So for this company, they went into complete response, and then they submitted an amendment. And they did something unique that we don't often see. They supplied data from their bioequivalence study to show that there was actually significant levels of this impurity as a metabolite. And it was present in both their test and reference samples. It was not a result of degradation, so it's not expected to go up over time. And the data on plasma levels was sufficient to qualify the impurity at the level of 3% because they're able to show that it was in much higher levels as a metabolite. So just in review, this was an ANDA for an oral drug product with a maximum daily dose of 1,000 milligrams. There's an impurity B that was exceeding qualification thresholds. The applicant submitted a metabolite justification using public literature. However, there was no quantitative information about systemic exposure, so they went into complete response. Ways to characterize safety with the metabolite justification: you can provide published literature with the quantitative information. You can conduct a non-clinical study, or you can look at your BE samples. So just in summary, the PharmTox team evaluates the safety of generics. The safety of impurity profiles are a major focus, but not the only focus of the PharmTox team. ICH and FDA guidances are key resources for us, and I showed you 2 case studies, the in silico prediction. Mutagenicity is good. Submit the 2 in silico methods in the full report. For general toxicity, we do not accept in silico prediction. Instead, submit repeat dose general toxicity studies or published literature. and do not rely on the Kramer classification to justify your limit. Metabolite justification to inform safety of impurity above ICH limits. Please provide data to demonstrate systemic exposure as a metabolite justifies level up.